This is the last video in the chemistry chapter. Woohoo! So we start with enzymes, and without enzymes we would have to heat our bodies up to a very high temperature to get reactions to take place. We can't do that because that will kill us. So in our body we use enzymes. They will speed up reactions. So the substrate or molecule that the enzyme acts on is called is the reactant in the reaction, but you need to know the term substrate. That is what the enzyme is going to act on. And the enzyme, being a protein, has a very, very specific shape. Remember that primary, secondary, quaternary, and tertiary shapes. So there is a specific spot on the enzyme that has a specific site shape for that substrate, and it's called the active site. So the enzyme and the substrate bind to form the enzyme-substrate complex. This interaction is very, very specific. Each enzyme has one specific substrate, and what the enzyme does is stress the substrate molecule, kind of tweak it a little bit, allowing a reaction to take place. And this reaction will just be the making or breaking of a chemical bond. Once the enzyme has made that change in the substrate, it releases it and it's free to act again. They may act thousands of times a minute. Some enzymes act 1,000,000 times a minute. They're very, very fast, very specific. There are not very many enzymes per cell, but there are many different types of enzyme. Because they're so fast, you don't need to have a lot of enzymes to act. So you can click on that video to see how enzymes work in the PowerPoint. So high energy compounds provide the energy for reactions to take place. They have a specific bond. It's a bond to a phosphate molecule. Phosphate is a group of ions, phosphorus and oxygen, and this bond to the phosphate is a high energy bond, and that energy basically is released when that bond is broken. So in our body, it is the molecule adenosine triphosphate. So it is I think of it as like a large clump with 3 phosphates coming off one side. And it is the very last phosphate bond that is the high energy bond. And we need this ATP for anything that requires energy in the body, whether it's building a protein, a carb, or lipid, whether it's contracting a muscle, whatever has to take place in the body requires ATP. It's considered to be the energy currency of life. So what happens is when a molecule is broken down, let's take glucose. When glucose is broken down in the mitochondria, it releases energy. That breakdown releases energy, and that energy is trapped in the high energy bond of this ADP, adding a phosphate to form a high energy bond, ATP. So this bond right here contains all that energy. So what does ATP stand for? You got it. Once the high energy bond in ATP is used, what remains? ADP. Remember, it's ADP and adds that high energy phosphate to form ATP, and it's reversible. So our last category are nucleic acids. You know your nucleic acids, DNA and RNA. They're rather large molecules. DNA is really large. RNA makes copies of the DNA, smaller copies. Carbon, hydrogen, oxygen, nitrogen, and phosphorus. And DNA is the information to make proteins. So DNA is considered to be a double helix because it forms 2 long chains that lie side by side and twist into a spiral. And it is made up of many, many units of nucleotides, billions of units of nucleotides. So DNA is deoxyribonucleic acid. RNA is ribonucleic acid. Both DNA and RNA are made of nucleotides. The nucleotide is made up of a base, a sugar, and a phosphate group. The bases are subdivided into purines and pyrimidines. The purines are larger. They're a double ring base. The pyrimidines are smaller, single ring. DNA and RNA differ in one base, whereas thymine is found in DNA. There is no thymine in RNA, just uracil. So if you ever see a strand of nucleotides with uracil, you know it's RNA. The sugar is deoxyribose in DNA and ribose in RNA, and the phosphate group is the same. So here's our structure. Phosphate group, sugar, and nitrogen-containing base. So here's a single ring structure. Okay, so the double rings, the purines, adenine and guanine, and the pyrimidines, cytosine, thymine, and uracil. So if you think of the DNA as a ladder that is twisted, the sides of the ladder are made up of the phosphate group and sugars bonded together, and that forms the backbone. The rungs of the ladder where you would step are the nitrogenous bases. So this is just representing the fact that the DNA is built by just about anything, by dehydration synthesis. So we take out a water in order to join two nucleotides together. Here's the phosphate group, the sugar, phosphate, sugar. So this is one nucleotide here with the phosphate, a sugar, and the nitrogen-containing base. DNA, as I said, is a double helix, and so it's two strands of nucleotides, and they're complementary. That means that adenine, or is represented by A, always pairs with thymine. And cytosine, represented by C, always pairs with guanine. Always. Now remember, in RNA, there is no thymine, there's uracil. So adenine always pairs with uracil in thymine. This is called complementary base pairing. And you can see this here, A with T, always. G with C, always. And these dots here represent hydrogen bonds that are holding the DNA, the nucleotides strands together to make the DNA molecule. If you were to heat that or use a particular enzyme, you would break those hydrogen bonds and open up those strands. So once those strands form, they twist together. RNA would be opening up the DNA and making a complementary copy of one chain. So RNA is different. It has a single chain. The sugar is different. It has ribose sugar. And one of the bases is different. It has uracil instead of thymine. RNA differs in other ways as well. DNA is stuck in the nucleus like reference books at the library. You can only make copies. And you know how the librarians are. You can't make copies of the whole book. You can only make partial copies. So DNA is stuck in the nucleus, and we make partial copies called RNA. Those can leave the nucleus, and the whole point of the RNA is to then go take the information from the DNA, take it out of the nucleus, go into the cell, and make proteins. And 3 types of RNA are required for that. Messenger RNA that carries the protein message, transfer RNA that is going to bring amino acids to the growing protein chain, and ribosomal RNA, which combines with proteins to make ribosomes. Ribosomes are the protein synthesizing factories of the cell. What are the components of a nucleotide? What is the monomer of nucleic acids? Hope you got that right. It's nucleotides. Which base is not found in DNA? Uracil.